Mrs. Rao is also called Tata. A middle name I cannot pronounce. Yeah, it's it's as complicated as mine. So please, I give word to you. Yeah, here you have a micro.
I have it, mine, and
But put you on here.
The same like you here. So
they told me I'm the only one who gets one.
No, as you said, it's like a unique microphone, to adjust it. But thank you, Peter. Good morning, everyone. Peter, thanks again for this kind invitation and to keeping me in the loop of this exciting meeting. To be honest, I learned a lot yesterday. And then on the top, actually, it kept my motivation now too high. And after talking with yesterday, some of you guys, it was very interesting discussion. I think we need to do a lot more research in MPN. That's the message I'm telling now myself. So what I thought of showing you some unpublished data from my laboratory. And before that, I give you again a little bit recap of fundamentals of MPN. Yesterday, we had a very nice didactic lecture from Laura. She introduced you what is MPN and how it evolves. So I go a little bit in that direction first few minutes, and then I go on and then discuss with you some unpublished and published data from my laboratory and my own experience. And I also a little bit touch why we failed to cure MPN so far and what needs to be done in the future. I add those stuff. So please feel free to interrupt me if you have any questions, and happy to answer you. Yes. All right. Just going back. These are my disclosures. The work I'm presenting today has nothing to do with my affiliation with my work with these companies. So I would like to start my talk by saying, or by asking you, how many of you like candies? How many of you like desserts? Can you please raise hands? Okay, I think it's not a bad number. So like the MPN cells do, right? So why I'm saying so, I will tell you why it's so important what you eat when you are healthy. But when you are ill, I think it's very important you should be mindful what you eat. Okay, so I learned and did one thing while preparing this presentation. Around 35% of all cancers are avoidable cancers are caused by diet. So this number is really means for me it's a huge, right? So it's in your hand actually how you can control your disease on daily basis. So I will a little bit touch why I'm saying this. So here I'm highlighting the diet. That is what you eat, what you are, right? So that is very important when you are healthy, but it's even more important when you have MPN, okay? So as Peter mentioned, I'm fully dedicated to find new ways, my career, find new ways to identify and target MPNs in a more meaningful way, if possible, to close to the curative approaches. So this cartoon you saw many times yesterday. So basically, MPN is not a single entity, right? It is it's a kind of, you classify like a caste, right, religion in subcaste. And so MPN is the same here. Based on the cell type increase, based on the symptoms, the patient show, scientists classified into 3 major flavors. One is P. vera, where you see more red blood cells. And the second entity, you have essential thrombocytosis, where megakaryocytes, more platelet production. And the third entity, we call it myelofibrosis. So the myelofibrosis can be primary. That means it's not coming via PV or ET, so it's a primary direction. And then you have other classification is secondary myelofibrosis, right? So that either come from the PV or ET can progress, that we call it secondary myelofibrosis. So both primary and secondary myelofibrosis basically share more or less same symptomatic features. There are a few tiny differences in terms of mutation burden, how fast they grow, and what was the previous history. But basically primary, secondary myelofibrosis, phenotypically similar to me. And as you know, we didn't talk a little bit yesterday, this part. So all these entities, PV, ET, myelofibrosis, can eventually, at a certain degree, depending on what additional mutation that patient acquired. For example, if patient get new mutation in P53, so the chances of getting and progression to secondary myelofibrosis or PVET to secondary acute leukemias. So that is the last stage, is the deadly stage, you can imagine. So all these entities actually basically arise. We talk a lot about it, and you are familiar with this because of changes in your gene, right? So those genes we already know. We talk a lot. It's one of the major genes that get mutated in MPN patients is JAK2. JAK2 kinase basically is the enzymatic activity it exerts in the cell, and that drive hematopoiesis in uncontrolled fashion. As Laura nicely alluded that part. I'm not going too much into that. And because of these mutations in JAK2 kinase, and I pointed out here two mutations, one of the major mutations we often talk, and many patients have it, almost 80-90%, is JAK2 V617F. So what does it mean, V617F? It's just a single nucleotide in your DNA change that leads to a valine replaced by phenylalanine. So phenylalanine, valine are amino acids that are actually building blocks of our protein. Now, because you have change in your gene, and then also you have change in the protein, and that change in the protein actually makes this protein structure so different. That structure makes this very constitutive, uncontrolled, without any trigger, your signaling, and then your hematopoiesis keep going. That's how you produce more blood cells. Okay. Now that is the major mutation, and subsequently, on the same JAK2 gene at a different place in the gene, people found mutation that is exon 12. Some patients, actually, these are not so many, very few patients, but this mutation exclusively drive erythropoiesis. No other phenotype patients, so no ET, no fibrosis. But it is a bit aggressive than exon 14, that is V617F. So biology is so interesting here. Same gene, two different mutations, two different locations. You have different phenotypes. And no one knows how different mutations on the same gene exerting different functions leading to different phenotypes. Okay, so that's one of the things biochemically we are trying to understand. If you know that why two different mutations cause on the same gene two different phenotypes, we can come in a new way to target those. Okay, so that's one thing. And then the second major mutation in 2013 by two independent groups, one in Vienna, one in Cambridge, identified this as the calreticulin. So the calreticulin, we heard, so by chemistry or biologically, it's a protein called chaperone protein. So the chaperones in our cells actually a kind of clearing or verifying mechanisms. If your protein is not so properly folded, it eliminates, basically. Now, this important protein is actually got mutated in patients, okay? Now, and these calreticulin mutations, actually two types. One is calreticulin 52. We say the type 1, when your doctor say, oh, you have CALR type 1 mutation, and you have insertion 5, that is type 2 mutation. Okay, again, there, not a hot spot one place. That gene is susceptible throughout, and then one mutation at deletion, other is insertion. But in this case, either deletion or insertion, both cause ET phenotype. Okay? I will a little bit talk why only ET but not PV if you have CALR, right? And then those CALR patients, ET patients, eventually can also progress to myelofibrosis. Okay? So again, all roads leads to the myelofibrosis, either directly or via ET or PV. Now, you have a third mutation called MPL. So MPL is the myeloproliferative leukemia, the name stands for, which is nothing but thrombopoietin receptor. So this is the gene actually present on your stem cells, blood-forming stem cells, sit on the surface, and then it waits for its partner. It's called thrombopoietin. Basically, it's a soluble circulating factor in your blood. And then when it touches this to the receptor on the stem cells, it activates the signaling, trigger megakaryopoiesis and platelet production. But now, because you have this mutation in MPL gene, and it is no longer required this stimulation via its partner, it itself can trigger alone. That's how you constantly, continuously produce, you end up having more platelets. Okay, both CALR and MPL cause essential thrombocytosis and myelofibrosis, but it won't touch much erythroid compartment. So the erythroid compartment mainly driven by JAK2 mutations. So that is the biology of this, okay? So what we are going to, so these mutations were identified, JAK2, almost 20 years ago, were identified. 2005, it was identified in Basel, Cambridge, and in Paris, and theoretically in 2013 and 2006 MPL. So why I'm telling these years? Because it's almost 20 years. We identified, we know which genetic modification drive these phenotypes, but we still don't have curative therapies, right? So it's a kind of a shame to scientists, for sure. We have to blame scientists and then clinicians. I think we are not going. I'm scientist. I take the blame. So one question, one possible reason I think why we fail to cure cancer, I think we are not looking at the right things, right direction, in the right time. Okay, so that's where we really have to invest. I will a little bit touch that part in my next slides. So this cartoon, it's my favorite cartoon, and because it came from my my excitement to do science in MPN. So what this cartoon I'm trying to show you, as I alluded before, that we are trying to find events that are changed early stage of your disease. To me, always as a scientist, late is too late. Okay, you need to you need to target your blood stem cells which are mutated in your bone marrow early enough. So what this cartoon is showing here to you is that, for example, we all have healthy blood stem cells. We call it hematopoietic stem cells in our bone marrow. When you are young and healthy, they function properly. And these are the cells, these are the precious cells are helping us to combat all infections and even cancers throughout our life, because they have two cardinal features. One is self-renewal capacity, meaning that they produce themselves like that, so that you have a pool reserve. And the same time with their second property called multi-differentiation property, meaning that these cells can give rise to all type of blood cells in your body throughout the life in a balanced, continuous fashion if you don't have mutation when you're young. Okay, so when you are aging, so you accumulate so much progressive changes in your body, inside the cell, outside the cell, including in your blood-forming stem cells. That already set a kind of a different tone compared to when you are young. So what happens when you are aged? You get more double platelets, even you don't have mutation. Okay, platelet number really goes high when you measure in your mother or in your child. Okay, so almost double. So that gave me some insights. We call it the myeloid bias. As the stem cells, when they get aged naturally, chronologically, they get a myeloid bias. Okay, so you can imagine why aged patient, if they have JAK2 mutation or CALR, the disease progression is really rapid compared to children. Okay, and we also modeling that in our mouse model. So we age mice when they are young and when they're aged, and we express this mutation, what we see in patients, and then we ask how kinetics of disease progression is really different or similar between young and aged patients. Okay, so but what I'm trying to show in this cartoon is that you have blood stem cells, and these blood stem cells by a bad luck or by accident you acquire at one point of your life. Right? So those mutations, I told you, JAK2, CALR, and then you also have additional mutations which are TET2D and MT3. So, and ASXL and so on. So the other mutations you don't hear very often. They are kind of set the base or a kind of secondary cooperating mutation. So these are epigenetic genes. So they do a different function than the JAK2 or calretically, but they also cooperate with their primary mutation. Okay, so those mutations you can also find when you do a next generation sequencing. Okay, now, so whatever reason you have your stem cell get mutated. Okay, so this stem cell mutated. So how long you think that you have a mutation and you get disease? Anyone knows how long it takes to get a disease? It depends, but my answer is it takes decades. Okay, so in other words, you are diagnosed with MPN today, probably those bad events happened 10 years ago, 20 years ago. Okay, so it takes decades, it's a long wait. Okay, so we are now also asking question, what is really driving from after having this mutation, okay? And why in some patients they never show. And the other thing you have to know is that this is no longer, to me, MPN is a rare disease, okay? Why I'm saying so? Because rare definition came 10, 20 years ago. That time we didn't have a good sensitive technology to identify how many mutant cells you have in your body. In other words, next generation sequencing was not so established. That time was very premature. Now, you can really fish out a single mutant stem cell or any cell, mature cell, from 10,000 healthy cells. Okay? So technology is so much improved. Now, with that technology, in Denmark, there's a big study. They measured almost 13,000 healthy volunteers to participate. Those who have cardiovascular disease, atherosclerosis, and other comorbidities, other problems, they all participated. They took the blood, they did the NGS sequence. So the surprise came. So what they found, out of 13,000, 635 patients have JAK2 V617F mutation. But those 635 never showed or never complained any or never diagnosed with a PV or ET or MPN. So that tells me is really that the number of patients with the mutation is more than the patients show the symptoms, okay? So there are two things. It's no longer rare disease. I think it's many people getting it, and no one knows why. JAK2, please.
Of course, there is a difference between having a mutation and having a disease.
Absolutely.
So although the mutation cannot be rare, the disease can still be rare.
Still be rare, exactly. That's what I wanted to say to you. Now, that is my next sentence, actually. So now you have more number of patients getting mutation, but not the same number showing the disease. So in other words, some people are blessed with overcoming the disease, right? Probably their immune system is good, their lifestyle is good, right? Or any other medication they are taking probably also helping. We don't know, right? But what you have to, the essence here, what you want to know, what you have to know is that getting this mutation is very often than getting the disease, okay? Now, if you know that why some people get disease, why others don't get, I think there are also some really important things you can learn, okay? As a scientist, I'm very much interested. Now, we know from no mutation to one single cell with mutation, and over time, decades, you end up in a diagnostic where you see a really detectable, from undetectable, from low number, very low number, to very high number, okay? In order to reach that stage, even though it takes decades, but this process requires a lot of energy, right?
In this study, they also search for the CAR. Sorry. In this study, they also search for the CAR mutation or just JAK2 mutation?
So in the first study, a very important question actually, the first study, proof of concept, they wanted to look how many people really have it. So they focused on JAK2, but now they also measuring in caloritically, general population.
Okay, thank you.
Mm-hmm. Now, so from an undetectable stage to a detectable stage, to reach that stage, you need a lot of energy to build those many cells, right? If you want to divide your cell, you have to give energy, otherwise it won't divide, right? So in other words, they really change their metabolism or the nutrient habits differently than when they are healthy, okay? So we think that to reach this process, as I mentioned, it takes decades. As I told you, this cartoon showing you, even some people have when they were born this mutation, okay, with this mutation. But they only show when they were 17 or 18. Yesterday we have heard 8-year-old, 17-year-old people have this. They probably acquired really when they are in mother or after the birth, okay? Now, how much time it takes, but what are the factors that really bringing them from this low stage to the high stage? If you know early enough, and this disease, I have to allude that this disease comes from the stem cells, okay? If you are mature, let's say your red blood cell get mutation, you don't show that lifelong, only 120 days red blood cells survive, okay? If your platelets get this mutation, let's say they survive only 8 to 10 days in your body because you constantly get new, right? So that cell is, but your stem cell get mutated. That is the problem we have. That's why we call this as a clonal disorder of hematopoiesis, right? Now, if a stem cell get mutated, we need to better understand at this stage, okay? So much of the research focused on you take the blood and you measure, you do sequencing, you do all biochemical assays, and also new therapies, for example, all pharma companies test on cell lines in the cell culture system. Yes, you put more drug, they die, it is very effective, right? But that's where I think we fail. I think we need to really understand, zoom, where the problem come from, right? You have to see where the seed is actually planted, how it is different from your healthy stem cell. Okay, that's where I'm focusing my laboratory. I would like to share our, some recent exciting findings at the end of my talk. And then also I kept the aging aspect, right? As we age, these mutations get, incidence of acquisition of this mutation is higher, and then also you have, on average, patients are more than 55, 60 years old. Okay, very rarely you find pediatric patients, we heard yesterday. Now, my laboratory asking if you know really what is happening, if you understand what nutrient or metabolic specificities of mutant stem cell versus healthy stem cell, what age-associated changes that are occurring inside the cell, outside the cell, you can go and attack those, and then and probably you can eliminate those stem cells. Okay, so that is what we are exactly doing in the lab. As Peter mentioned, that you see a lot of dead mice and dead things. Don't be scary to see those pictures, but I will tell you also how important it is to model first before you go into patients. And the mouse model is not like a tiny furry human, but they're kind of useful models. I will show that. But before that, I would like to show you one cartoon. So what you see in this cycle, so it is a very famous publication from Douglas Hanhanen from Lausanne. So he repeatedly pointing the characteristics of cancer cells. So we call it hallmarks of cancer cells, okay? So these properties, what was written on this circle, are entirely different from your healthy stem cells, okay? Now, so two I highlighted, we talked a lot about it, sorry. Yeah, we talked a lot about it is and the metabolism, so, and then also the inflammation, right? MPN is intimately associated with inflammation and metabolic changes. So these two properties are already considered as one of the hallmarks of cancer cells, okay? So my laboratory also studying how these two hallmarks of cancer cells talk to each other and then further drive the disease. Okay, so that's also, I will a little bit show you today the data that what we learned from studying these two properties. So as I mentioned, we first test our ideas, hypothesis, what is different between healthy and mutant stem cell using mouse models. So these mouse models were generated by Radek Skoda laboratory in Basel, where I initially trained on this disease, and then also others, where they engineered these mouse models by putting the mutation, what you see in patients, into the stem cell, into these mice. And there you can really controllable way, you can activate these mutations. Okay, they engineered such a way. And then you can appreciate here that this mouse model, like in patients, in the blue curve and red curve, those are the JAK2 mutation expressing mouse models, compared to the black bar is the wild type, so no mutation. Okay, so like in patient, you see a very big spleen within no time. These mice they show disease bit rapid than patients because their 100% cells are mutant. That's why you have a bit more rapid kinetic. Okay, in patients you have 20%, 2%, depending on what stage you have it. Okay, that differs the kinetic of your disease. Now, you see the splenomegaly increased, red blood cell number increased, platelet increased, and then also they have a poor survival if you compare to the black curve to the red and blue, which are JAK2 mutant mice. Okay, so this cartoon tells you that mouse models in a way are very useful. They nicely mimic the patient situation, what you see, for example, splenomegaly, blood counts, and survival. Now, so use those models, we started asking our questions, what is the role of altered metabolism in MPN pathogenesis? Okay, so what you see, this picture is not so nice to see it, but that's how it looks if you open these mice. Okay, the first mice, what you see is the black one is the wild type mice, no mutation. They have a smaller spleen. I highlighted with the red box. But if you have a mutation, you see how the spleen looks like. Okay, so in both models, over time, it's 10 times bigger. It pushes the lung and heart and then uncomfort feeling, right? But what we learned, in addition to they develop these classical MPN symptoms, and I also highlighted with blue and red stars. If you focus there, so compared to healthy mice, where healthy mice have a nice fat distribution under the skin, we call subcutaneous fat, and then they have a gonadal fat above in the belly fat, nicely. But if you activate mutations, this fat is really, really disappeared, resolved over time. And if you see the same picture, same mice, after 2 weeks or 3 weeks or a bit more time when we wait, and then even you don't see the fat. So we call it body weight loss or fatigue or the cachexia phenotype, we call it, right? So these most models also show such phenotypes, right? And then, but we were really wondered, how come we activated mutation in the blood stem cells, but we have problem in the systemic whole body, right? So how this is happening, and what is the really mediator or the secreted factors that come from your mutant blood cells and also attacking other non-hematopoietic tissues, in this case, the muscle wasting, fat loss, okay? So we systematically studied this aspect when after seeing these differences, you see now blue curve, red curve, the body weight. So when we measure body weight of these mice compared to mice with no mutation, they started falling apart. And then what we also found actually in these models, in the next curve here, showing glucose levels. Okay, when you measure the blood glucose levels, mice with the severe PV stage, late stage, your blood glucose levels really going down. Okay, and this holds true in patients if you measure glycosylated hemoglobin. So that is the indication of appropriate glucose levels in the blood. So in the severe PV stage, if you have 5 to 10 times more red blood cells than when you are healthy, so you get this hypoglycemic phenotype. And that was very interesting, and I will talk a little bit that. So that explains why people get fatigue. So why I'm saying fatigue here now, because red blood cells like so much of glucose, okay? They don't have a nucleus in their cell because when they mature from stem cell to the red blood cell, they spit out their nucleus, okay? Unlike other cells in the body. And they heavily rely on glycolysis metabolism, which is fueled by glucose, okay? So now you can imagine you have 5 times, 10 times more red blood cells, and you eat the same amount, but you have cells which are more dependent on glucose consume all the glucose, and you have no glucose available for your heart, for your muscle, for your brain, and you can really think now, okay, I'm not feeling good. That could explain why, biochemically, why you get fatigue, why you feel tired so fast. And if that continuously happening, and you also provoke other changes to accommodate or to compensate those problems, okay? And then the additional problems come. But over time, right? Now, we found that red blood cell use more glucose, and those glucose is not sufficiently available for your other tissues, which heavily depend on, including brain and muscle, and that's how one get fatigue, which we think. Okay, so that it's not only in mouse models. I come to the data of the patient data that we and others gathered. So then I ask question whether diet, what we eat, does it make a difference on MPN pathogenesis overall and also day-to-day basis, okay? And that also we model by exposing our mice. I told you before, mice with MPN mutations, they lose body weight. And then I asked, can we rescue this loss of body weight cachexia phenotype by feeding them high-fat diet? Basically, you eat more cheese and more bad food, okay? That's what we exposed to mice. And then you see now, compared to in the previous slide, that mice got a balanced diet we call chow diet in the research, and that has normal carbohydrates and fat and other minerals in balanced way. But when we expose the same wild-type mice with high-fat diet, you see they become really chubby. That's what you expect, right? That's what's happening. But the same diet when we expose to MPN, you see now what's the picture. You can easily compare. Yes, they did gain little weight, but compared to healthy mice, they didn't gain weight, right? The more you provide, you feed the beast in you, okay? So that's what's happening. And you still have spleen size is the same, bigger, even bigger, right? Right? So now, you see, it's not only in male or female. We ask whether these differences are only specific to female or male. We systematically went and then checked the gender differences, and it holds true in both cases. But in mice, males do bit different than humans. Okay, so it's a kind of evolutionarily, there is some difference between mice and human in terms of gender when you look metabolic aspect. But overall, take-home message is that it's true for both male and female. Okay. Now, after seeing this, we also ask, okay, overall picture look like. What about blood counts? So here comes the surprise that when we expose mice with the food that has more than 85% fat, okay, that's what we did. And then in this case, we model calreticulin, mice expressing calreticulin mutation, and then we measured blood counts, including red blood cells, platelets, white blood cells. Since calreticulin mutation drives exclusively platelet phenotype, you can see within no time, platelets so increased when you expose them to high-fat diet. Compare this red curve with high-fat diet, black curve with balanced diet. So, and then we really went on and then went to the details why this is happening at the cellular level, biochemical level. I can tell you in one word that imagine a big cell megakaryocyte. One megakaryocyte on average in humans can give rise to 600 platelets on average. Okay, and the platelets are nothing but the clones, they shed from like a balloon, small balloons, packed vesicles from megakaryocytes. Okay, and that number heavily increase when you expose to high-fat diet because these platelets are really packed with fat. They are just membrane, and this membrane is full of made with fat biochemically. Now, so you provide more fat, they utilize it to produce those tiny balls, and then you increase. So this is one tiny information from these small preclinical models of MPN. You see how even just, they have the same mutation, you just expose them to different diet, and then it fits. Okay, so this is one example how important it is. You think if you have PET, what you should eat, what you should. I'm not a cancer nutritionist. I should not advise anything, but I advise you to consult your cancer nutritionist. He or she can advise together with your physician. Okay. Now, you might be wondering, Rao, I have PV, I have JAK2 mutation. So what I should eat it? As you know that PV patients have more red blood cells, 3 to 4, 5 times more red blood cells. As I told you, red blood cells like glucose, like we all. Okay. Now they like even more, but they not only use more, they also use differently. That's also we learned. Okay. I'm not going to that part, but if you have any questions, if you need more information, I will be happy to provide. Now, in this case, so we switch to JAK2 model here, right? JAK2 models, I told you, they exclusively show PV phenotype, red blood cells. They have 5 times more red blood cells. Now, we expose them to 8 to 12 weeks with a normal diet, but in the other group, we expose to high glucose diet. So it's more than 65% carbohydrate, it's a fructose sugar, sucrose or glucose, the composition, whatever carbohydrate, that in the end, it leads to the glucose. Glucose is utilized, your cell taken up. And then you can see now in the pictures there, the first bar graph, what is showing, black corresponds to wild type no mutation. They have no problem. They have a balanced way to utilize it. They don't utilize too much, right? But now it came to the surprise, even at stem cell level, we found when we exposed the JAK2 model with high glucose diet, stem cell number went down, okay? Number went down transiently, but that number, they went down because they're pushing towards the erythrocytes, erythropoiesis. That's why the next subsequent stage, where I labeled it MPP, multipotent progenitor, that is one step down of your hematopoietic stem cell. If you put a hierarchy, stem cells stays first, and the next step is MPP, that is the uncommitted. And then you have, I call it pre-CFUE, these are scientific terms, colony forming unit erythroid. So basically that is the progenitor cell, stem cell, destined or decided to go to erythropoiesis. No longer it has a multipotency. It can no longer give other cell type. Okay? Now, if you look at here, it start picking up. Already at stem cell number, within no time, 8 weeks, 12 weeks. Okay, imagine patient constantly taking too much dessert, too much sucre, too much beverages, over time 10 years. Slowly, every day, it pushes erythropoiesis, slowly. That's what you see, it progressive changes. Now, when we also measure reticulocytes. Reticulocytes are nothing but one stage of the mature red blood cells. They are just freshly generated. They're not old 120 days living, just came from stem cells. Those are reticulocytes, which you can also measure in your diagnosis. You see again, IFAD diet really picking up, even within no time. So that's scary to me, that, okay, you feed your cells whatever they want, and then you complain that I have more disease, my treatment not working. Okay, treatment is one side, but also something is in your hand. What you eat daily, I think you should really think what disease you have it. This information I'm giving everything from mouse models that we gathered. Okay. Yes, please.
In this case, you're focusing on exon 12?
We also did on JAK2 V617F as well.
And that's the same result?
Similar trend.
Okay.
Similar trend. JAK2 exon 12 show a bit more faster kinetics, but just for presentation I picked, but we have all data on JAK2 V617F as well. Now, so I just wanted to show you a little bit how we can take this information for a therapeutic purpose. In other words, I ask a question, is there any therapeutic window on metabolism-based approaches? Okay, based on our findings and findings from other people in the field. So for that, we did a very detailed molecular analysis. So what you see, the form of heat map here, we call it. Yesterday, Ruben nicely alluded that similar cartoons, where you have blue is low gene expression, red is high gene expression. Okay, where we took the stem cells from the mice with no mutation, mice with the JAK2 mutation, and we systematically look the molecular changes by a technique called RNA sequencing, which is similar to next generation sequencing you do for mutation profiling. Okay, now, and then we listed out the genes, what you see, those PKM, PKFB3, those all genes are very important in our normal cells, but they are heavily upregulated. In blue and red, you see E12 and V617F. If you look compared to wild type, blue means no expression or very basal expression, which we needed, but they massively upregulated in the red, right? So that facilitates, that means after expressing mutation, your cells are changing molecularly something, and that facilitates to use more glucose, okay, more glycolysis. And then after seeing that such a nice pattern, we focused on, can we use this information to target MPN cell growth, to reduce platelets, or to reduce spleen size, to reduce erythropoiesis. So in this project, what we looked into it, one of the genes that caught our attention is PFKFB3. So this is the rate-limiting gene of the glycolysis. Basically, what it does is that it speeds up your glucose utilization. Okay, if you have more of this enzyme in your mutant cell, it speeds up the glucose process to the end product. That's how they get energy faster. So that's what I meant. They not only take more, but they also utilize really differently because of this molecular wiring. Now, after all our efforts, that led to identification of some important genes, PFKFB3. So fortunately, there is a drug that can inhibit the activity of this molecule. Okay? As a proof of concept, in mouse models, we tested what if you inhibit the activity of this differently present enzyme with this drug, can we modulate the disease progression? Okay, that's what exactly we modeled. I showed you in the cartoon here. So we exposed the same drug with no mutation, with mutations, mutation 1 and 2. So blue graph showing the spleen size, okay? So what we did now, we didn't treat V617F red in the exon 12 mice with no treatment. We call vehicle, it's a placebo, okay? Now, and then we treated this with this drug V1, Warburg effector inhibitor 1, we call it. It's for historical reason. And Warburg is the guy who actually found cancer cells utilize glucose. Even 100 years ago, without any of these tools, fancy tools. Okay, this was forgotten almost 80 years. Now, since the last 20 years, again, people dig the literature, and then actually, holy people identified this important information a long time ago. Okay, now people focusing on this, including us. Now, you can appreciate here, when you treat with our inhibitor in JAK2 models, you can really reduce within 8 weeks. We didn't treat too long, but if we treat too long, we can really maintain it. It is tolerable if you reduce the glucose utilization with this inhibitor, but you can see that spleen size is really reduced in both models. And we also combine that with the classical JAK inhibitor, if it can further make it better, okay, in a combination therapy. Okay, so in exon 12 model, it is very striking. In the last red curves, you see it. When we combined our inhibitor with JAK inhibitor, Ruxo, the spleen size is really getting back to the normal size. Okay, so that was very exciting to us, that our hypothesis that indeed metabolism really playing a role in driving disease, and inhibiting metabolism, you can also alter the disease progression. So you might be wondering, Rao, metabolism is important for all normal cells, normal function, glucose also important. How can you target in patients? You probably end up with side effects. Okay, time will tell, but my answer is here, you are going with a specific target that is not expressed in your hematopoiesis. If you look at this gene that I put the arrow here, it is not expressed PFKFB3 in your normal healthy cell. Okay? It is only turned on if you have mutation. So I call it, they are really a specific targets. Okay? How much it is specific, time will tell. We need to do more work. Okay? And then with the approach, we don't want to really completely inhibit. We want to bring back to our healthy level. That's it. Okay? I'm not here saying not to completely to make it zero. Yes, if you make it zero, other cells also get affected, right? You bring back to, you tone it down. So when you tone it down, even mutant cells no longer have the fuel to proliferate, to differentiate. MPN is nothing but a number game, right? We talk a lot. Okay, I have 500, 600, 13, 20, right? Depending on what parameter you're looking. So this number game is fueled by what you eat. That's what I'm trying to say here. So be mindful what you eat, which disease you have. So then I was very satisfying to see our data was really confirmed, mouse data, what I showed you, by people like Ruben Mesa, actually, this study from one of his colleagues, and he in his department, what they showed 1 year after we did the mouse work, they exactly came with a very nice data from inpatient cohort. What they elegantly did, they took a cohort called NIH AARP diet cohort. That was a very big study in the United States, recruited 450,000 volunteers. And then they they did a questionnaire, what they eat, how much they eat, and in that study, systematic study, out of this number, they found 500 people are with MPN. Okay, out of these 450 people. Now, these 150 of them ET, 190 of them PV, and others probably myelofibrotic. Okay. Now, you see, general population, already you have a 500 MPN. And then what they did, they classified based on how much carbohydrate they eat, in sugars they eat on daily basis. So here comes the surprise. So very satisfying to see that what we found exactly in mouse models, they did very unbiased study here they found. So what this cartoon shows here, maybe I go a bit closer. So here, this line tells the patients with 70 grams per day sugar they took, okay? And then these patients actually above 120, okay, within MPN patients, 120 grams versus 70 grams. So low sugar, high sugar, okay? Now, here the statistics telling you hazard ratio, okay? So hazard ratio here, very significant, means the disease chances of a disease progression is higher. And then the phenotype they show is very higher if they took the more than 117 gram versus 70 grams. So then you might be wondering, Rao, sugar comes from what? Chocolate, milk, fruits, right? All contain sugar, right? They also systematically looked, okay, whether how much fruit they take it, whether sugar come from natural source, sugar come from beverages, or the fruits fructose, okay? Irrespective where it come from. They found a statistical, meaningful, a significant way that if they take more sugar, they have more chance of increasing the disease progression higher. So, okay. So this is again a very nice comparison. We looked into mouse models, and then they looked into patients in unbiased way. Two independent studies. No one knows each other what they are doing that time, but they come to the same conclusion. Okay. So in other words, you really need to think what you eat when you have a PV, ET, or myelofibrosis. Now. So, and what about fasting, So during festivals, because of religious festivals or ancient people, actually, they did the fasting without knowing its benefit. But they probably felt benefit over time to lose body weight or to clear all your bad things in your body. And we also exposed our mice with a fasting, meaning that we restrict the calories there with the food. In the food we designed that is less calories, and those less calorie diet, we put them 12 hours food, 12 hours no food, not continuous exposure. So we did several cycles like this, and then we ask how it affects. Here comes the surprise. Again, so your JAK2 model we used, and then the dotted lines in the red you focus, that is with fasting, so food on and off. And then the continuous line, the red line, it's a continuously we fed. Okay, that's what we do regularly. And you see the difference, mice with the same mutation, we have fasting. Okay, less nutrition availability, less disease progression. But again, you might be wondering, Rao, I one side complain fatigue, I have no energy. Now you suggest me that fasting is useful, right? Again, so it depends on what you eat, not that how much you eat and what you eat, I'm trying to say. Okay, you should not completely not eat. Okay, you should eat to maintain your healthy routine. But what you eat, how much you eat, it is very important. Okay, now it not only reduced blood counts, but it also reduced spleen size. That was very surprising, because all increased blood cells, you have a bigger spleen, right? It was a kind of fitting. Now, I just wanted to summarize what I told you, right, in this cartoon. So here you have stem cells. It gets mutated. Mutation leading to molecular changes, including changes in your glucose transporters into your cell. Okay, any nutrition, glucose, fat that enter to your cell via receptors present on the cell. Okay, I highlighted in the blue. And their level of expression increase if you have mutation because of molecular change. Together, they work together, they take up more glucose, and that energy facilitates to produce more blood cells. Okay? Now, what happens if you continuously utilize your money? You don't have any more money, right? You depend on bank to get some debit, right? Bank is a fat here in your body, right? So that is exactly what is happening. I showed you in my first cartoon that mice even lose body weight. They lose fat. So what happens that when they reach to produce a lot of cells over time, they don't have any more energy. If you don't eat it, you don't provide. They search for the alternative source, which is fat, right? So that's why fat becomes shrinking. So you lose over time the fat, okay? So that fat is you lost, and then at the end you don't have fat and you lose the body weight, right? And also eventually you lose muscle weight, right? That's where the real cachexia phenotype comes. So how do they do it? How mutant cells go and contact bank to get the money, in this case fat. So they secrete some inflammatory cytokines. So again, inflammation come into play a role. So how inflammation, we talk a lot about inflammation increase in MPN patient. What it really does, right? This is one of the things it does, right? It go and stinch fat release, it's a stored energy. Fat is nothing but full of energy storage, right? That they release, and then your cells utilize. And if that continuously happening, your bigger fat cell becomes smaller fat cell, right? And then you lose the fat. That's how you get the cachexia phenotype. That's how you lose the body weight in cancer situation, including in MPN. And this happens more in severe PV and severe myelofibrosis models. We also notice that, and this holds true in patients. Okay? Now, so this is the mechanism. Any questions so far, if anything? Okay, so if we can also discuss later if you have any questions. So what I'm going to tell you now, how we take further, how we take further this information to make it more beneficial to the patients, okay? So the cartoons I'm going to show you next 3 slides I got from my good friend of mine, Angela Fleischman. So she's also clinician. She worked a lot on MPN. She's California-based researcher, clinician. And what she did after studying our research, publication also from Ruben Mesa, and she initiated a program. She recruited almost 50 MPN patients, and then now she wanted to keep them on a controlled diet, like we did for mice, okay? Now, and before that, I like to show you this publication, landmark paper, that came from actually Spain, okay? Valencia, Barcelona, Madrid, and all collaborators did on cardiovascular disease, okay? As we know that if you have heart problem, you always think about fat, right? Why can't we think the same in MPN? Right? We now know that fat feed platelets, glucose feed erythropoiesis. Something else feeds myelofibrosis, okay, which we are working. Now, so this paper really stimulated many researchers, other cancer researchers, to do the same thing in different settings. So what they did, they simply exposed patients with Mediterranean diet with compared to Western diet or their natural diet. Okay. Now they found really good beneficial effects if patients under Mediterranean diet. That's what Angela Fleischman did. So what is Mediterranean diet? You guys know better than me, right? So it is basically, if I define that, it's a simple plant-based diet, balanced diet, fiber-rich, and it exerts anti-inflammatory properties. Basically, it reduces inflammation. Because of more fibers, then you ask how fibers can help my metabolism. It gives the beneficial effect. The fibers, basically, when you take, it kind of put a layer in your stomach, okay? And that layer is important for the microbiota, what you have in your gut, in your stomach. And also, it slows down the glucose uptake. Okay, even if you take the same amount, which is not recommended, because it is coated with fibers inside the stomach, and it slows down the absorption of your glucose. It's not pushing so quickly. Okay, and that trigger is slowed down. You are not hungry, and then also you feed your cells a bit controlled way. Okay, that's how fiber-rich diet helps. Okay, one way to microbiota, one way to slow down the utilization of your sugars. Okay, now the similar test. This is the regime Angela Fleischman did in her clinical cohort. She recruited 31 patients in the first study, and now she increased to up to 50. She divided them 50-50. She exposed them to Mediterranean diet and the US diet, I would say, Western diet. Now, here it comes the surprise. What she found here, very interestingly, she followed up to 15 weeks these patients, and they really strict to adhere to our suggestions. They did exactly what she asked to eat. And then she compared the black bars or the no control. They can eat what they want. But in the pink bars, you see that it's really falling part of the signature MPN SAP score. So yesterday, Laura and Ruben nicely alluded the SAP score symptoms. So how you are feeling today, whether you have any bad mood, whether you're social, anxiety, fear, night sweats, all those stuff they counted, and then they found over time, compared to normal diet people, 17 versus 47, 31 versus 53. These looks like a tiny difference. You know how much difference you need to feel bad and good, right? It's getting to the positive side. Why can't you use that in the form of diet, and then the top you add yoga, meditation, and other good things along with your medication. I think we are getting there. We are not there yet, but we need studies like this more in different places to see how reliable it is, how useful it is, and then we can really find new ways to modulate the disease. Now, so this is the study from Angela Fleischman lab, and we talk a lot about thrombosis. Martin yesterday nicely alluded, so how it evolved thrombosis when your platelet gets activated. Thrombosis comes, right? And in ET patients, you not only have more platelets, but they're also very sensitive to if you give any trigger in the form of collagen exposure or DNA exposure or inflammatory exposure, when they touch to neutrophils, they kind of activated, they release this. factors. That's how you get it. So, but what she also found that she connected to metabolism. So MPN cells use more metabolism. What happens if you use more? You produce more waste, right? And one of the waste in our cell is called reactive oxygen species. So those are oxygen molecules like a fine sharp needles, and they can go and attack all the parts of our cell, components of our cell. Okay, that's how they cause the activation of platelets. If that is happening in platelets, that provoke the thrombosis. And then what she found that, so she came up with the antioxidant, which is called N-acetylcysteine, which is the very old drug. Basically, this drug reduces the ROS levels, basically, okay? That's, if you reduce ROS levels, okay, even if you have more metabolism, you can reduce thrombosis. Okay, that is very nice study in vitro, in the cell culture models, in mouse models. Now it needs to be tested in patients. Okay, so why I'm bringing this? How important it is that metabolism actually the major culprit. Okay, to produce more number of cells, and then you have more cells. You always have to think your number. This number coming because you're feeding it. Okay, now. So I switched a little bit my gears to a new thing. So Peter asked me to also a little bit touch what is new in MPN. Okay, so what I told you is the past, and that is the information I shared with you from our data colleagues from in the field. But now what is new? What else we can bring and what is missing? Okay, I'm going to share with you. It is a bit confidential. Please don't share with anyone. This part of this study we now submitted to a manuscript in Nature Cancer. So two questions I ask as a scientist, okay, why and how. So in this case, how your mutations expressing in blood stem cell causing entire problem, body problem, systemic problem. So what are the factors that connecting blood stem cells to the systemic part? And then second, I ask, can the systemic alteration can be harnessed? Can we leverage those changes, those findings to find new ways to target? And even to find the biomarkers. So yesterday, our discussions, Ruben Mesa, he also mentioned we really don't have a good biomarkers. Okay, and also one of the MF patients, they said that they were wrongly diagnosed with ET, even though that patient have MF. It's such a sad thing. So still we don't know how to distinguish in an easy way and in a reliable way. Okay, so that part I'm going to touch now. So very exciting data we discovered for last work from last 4 years. Very extensive work. This cartoon showing. So what I did here, I no longer stick to mouse models. Now I have a very big access to MPN patients. Okay, so patient plasma or blood cells or bone marrow aspirate. So you can see the number, actually, the number of patients that I have access now. I collaborated with different teams, basically, okay, all over the world. And together with our mouse models, combining these two important tools, and I did all possible molecular biochemical assays, okay, to really distinguish what is similar between mouse models and MPN patients, and how they are different between when they are healthy, okay? So the ultimate goal is to find new targets, new biomarkers, and then use them for therapeutic target. So I'm going to share with you all our experts led to more than 1,500 reliable candidates. We cannot study all 1,500. We have to focus on few things, and then we focused on for several reasons. I'm going to show you a factor called GDF15. So GDF15 stands for growth differentiation factor 15. So it's kind of one of the inflammatory molecule. You can put it in simple words. So it circulates in your blood. So what we found that this molecule, basically, both in mouse models and patients, is one of the really reliable, faithful candidate, is very heavily upregulated. If you look at the graphs, it's the time kinetics. We measured in the blood of this molecule, of the red and blue or JAK2, black is wild type, no mutation. As the disease progresses, is speaking up. Okay, 10 times more in exantol. Calreticulin is the same, exactly. I didn't show in this cartoon. I need to update for presentation. But again, in the circulation, you have so much GDF15 in mouse model that anti-correlated with the body weight. You see the next graph showing that if you have more GDF15, you have less body weight. Okay? So we have a factor now that defines why patients lose body weight, okay? Why they lose cachexia. Now, measuring those, and then you might be wondering, Rao, show me the data in patients, right? So that's what we did exactly, the lower group here. The first bar showing the healthy controls. We have 55 healthy controls who has no hematological symptoms, no MPN, no other blood problems, okay? Now, and we looked into PVET, secondary myelofibrosis, primary myelofibrosis, even triple negative patients, a large number. You see, not like in mouse models, in patients you have a continuum, but you have a really significant and 10 times higher increase of GDF15 in their blood. Okay, this was very surprising to this result to see us even in patients. And then what we found more interestingly that myelofibrotic patients is way high because it is progressively accumulating. It makes sense, right? Myelofibrosis, you call it kind of after the PVET or at the late stage, or even those cells produce more. But at the end, myelofibrotic patients have so much higher, 10 times more higher, and that gave me these results gave me one question, Rao, can you use this to find myelofibrotic progression at early stage? And then, in other words, I asked, can we use this as a biomarker to predict myelofibrosis evolution? Okay, and then that consumed a lot of my time.
I expect that those patients you have as reference group also have medicine.
Absolutely.
Yeah.
Why you see?
What is the effect there? Yeah.
Absolutely. We also extensively did how ruxolitinib effect, how interferon effect. We have all this data. For the sake of time limit, I'm not going. If you are interested, I will be happy to discuss that part. Yes. So one question we asked, this increased GDF15, it can be used as a biomarker to predict myelofibrosis. As you know, myelofibrosis is a diagnosis very painful. You go with thick needle, you insert here, you aspirate it, and the chances of getting the bone marrow into your needle is even low if you have fibrosis, because your tissue is not filled with the marrow anymore, right? You can get it from PVET, but in myelofibrotic. So that painful procedure, and also we don't know when patient actually progressing to myelofibrosis, okay? I thought it's very important to understand using GDF15 as a biomarker, and we did some computation stimulation. So what you see here is the curves. In the clinic we do these curves is called receiver operating characteristic curves, ROC curves. Whenever you heard this term, that means you are defining the specificity, sensitivity of a marker that you identified. How specific it is to your disease, how sensitive it is to predict as a biomarker, okay? Now, that's what we did. Upper is primary myelofibrosis, lower is secondary myelofibrosis patient group. And then what we did, we set a base 190 picogram that you find in healthy people, okay? And then asking question how this curve falls, okay? And if the curve is really straight like you see it here, we call it area under curve, AUC, when it is close to 1. And it gave very good sensitivity of 94% in PMF, and even secondary myelofibrosis, 100%. Okay? So this gave us a confidence that we can really use as a predictive biomarker when ET or PV patient progress to myelofibrosis. Okay? All you have to do is not the bone marrow, maybe bone marrow in the first case, but take the blood. It's very easy, right? Instead of bone marrow. And then you run this assay to measure GDF15. Okay? Then you can really measure it. Now, these are computational predictions. Okay? Now, Rao, show me the data, whether it is true. So, and then we went on and collaborated with in Canada and in Austria. Same patient when they were at ET or PV. So here I'm showing out of 33 patients we have PV, patient 1, patient 2. When they are at PV or ET, when they transform to the myelofibrosis after 3 or 5 years later, we also have access to the bone marrow from the same patient. Okay? And then we simply did expression analysis for GDF15 by a technique called immunohistochemistry. That's what people do in pathology and diagnosis. And then what you see in the last, I don't know whether it's good on the screen, but red dots on the last part, okay? Last stage. So that is corresponds to the GDF15 expression in bone marrow. Out of 33, all 33 patients, irrespective whether they are Ruxo or interferon, when they progress, they have this higher expression of GDF15. So this gave us a confidence that you can indeed faithfully use GDF15 as a non-invasive. I'm saying that no needle insertion is required anymore. You take the blood, you measure. Of course, you have to rely also on other parameters what we have, and then combine those to get a nice conclusion, not to get confusion whether it's ET or it's MF. And the other advantage of doing this, having these biomarkers, clinician can also decide and adjust the dose or the treatment type, okay? So that's also, this is very, very useful information. And then I would like to tell you two other exciting in the next two slides, where the GDF15 is just a cause or consequence, because you have more mutant cells, you have more production of GDF15. And then we also ask that question, whether it is a consequence or a cause, okay? Whether, in other words, it's also promoting the disease. Okay? For that, we switch back to our mouse models. They are very useful, where you can modulate a lot. You cannot do anything in patients at this stage. And where we did was we genetically knocked out. So in these mice, you have mutation of JAK2, but no GDF15. Okay, and ask question, how disease progress in these models without GDF15. And you can see in the cartoon body weight, I told you, JAK2 exon 12 mice, for example, severely aggressive disease, it leads to body weight reduction. When they have JAK2, you lose body weight compared to a black curve here, wild type, no mutation. But here in the cyan color, you see it, JAK2 model without GDF15, it even increased the body weight, right? You really can reduce the cachexia phenotype that is coming from you because of your MPN mutation. This is very, very striking. Now, what about hematological parameters? Indeed, actually, it also reduced spleen weight within 6 to 8 weeks after removing, and it even improved the survival. It extended, when the mice have the JAK2 exon, they can only survive 50 days. You can extend to 75 days. So that's a very good improvement. Now, you ask, okay, I don't have JAK2 exon 12 RAW, but I have JAK2 V617F. What about JAK2 V617? So we went on, also did, we knocked out the gene. It takes 1.5 years. This whole cartoon work took almost 1.5 years, okay? Because to genetically manipulate, your mice should have JAK2 V617F mutation, same time it should not have a GDF15. So this technically takes even more than 1.5 years. But it was worth, you can appreciate here now, when we knock out GDF15, surprisingly in JAK2 models, it didn't touch much of the erythroid compartment. It really lowered the myeloid compartment, where neutrophils, white blood cells, monocytes, you see the blue versus red curves in the bottom panel, really reduced. But very strikingly, what we found is that the platelet number on the top right there is really blue versus red. Blue is with mutation with more GDF. Red is with mutation but no GDF15. Okay? The platelet number almost 50% reduced within 20 weeks when we followed these mice. Okay? That is very striking. I never saw any drug reduce that much, including Jakavi or interferon. Now, okay, Rao, I have a calreticulin. What should I do? Right? And that's also we did. That again, in parallel, we knocked out in calreticulin. We got the same picture for platelets. And it has no effect on, anyway, CALR people don't show any red blood cell phenotype. That makes sense. And then the myeloid compartment didn't get affected, but platelets again. Really 50% reduced. It's not just in one model. And in all models we assessed, it's genetically, it's very conclusive data. We think this could be used as a not only biomarker. In future, we should really inhibit this molecule in MPN patients. So that's where I took a very big step, invested a lot of time to develop an antibody to target GDF15. Because you have too much, I want to inhibit that in patients. Okay, so this is the antibody we generated. We tested in mouse models both calreticulin and JAK2 V617F, where we injected human patient-derived cells into mice. We call it xenograft, and where we followed systematically with our antibody, without antibody. So you compare now for color, for example, pink versus cyan. Okay, within 4 weeks, these cells really grown so much in these mice, human cells, whereas when we treat the same mice with the same cell with antibody, you can really reduce. That's exactly what we found in mouse-to-mouse experiment. So that means this, indeed, you can pharmacologically inhibit your GDF15 in your body with antibody or a different pharmacological approach. We are also now currently pursuing it with a different approach, not only antibody. Okay, so but this is unpublished. We recently submitted to manuscript, and hope reviewers see the same potential that we see it. Now, so one final or two final cartoons I would like to show you. Peter asked me, Rao, so which direction we should MPN research go? Okay, so what else missing? To my knowledge, to my understanding, we know nothing about the true things happening at the stem cell. We are just started, I think. We need to do a lot, as we discussed yesterday. So that explains why we are failing to cure cancer. Okay, I would like to give 2 slides insights why we are failing to cure cancer. So one problem is resistance. Okay, resistance, like if you give antibiotic, is no longer working because your bacteria is not responding to that. Same likewise here, your cancer cells not responding to your Ruxo or interferon. Okay, but that also after prolonged treatment. Okay, resistant is one problem, and then other problem is you have to look carefully. We are talking about JAK2, calreticulin, MPL. All these genes, when you are healthy, they are very important genes to keep your hematopoiesis ongoing. Okay, now such important genes get mutated. Okay, now you want to target those mutant genes, right? With ruxolitinib is nothing but JAK inhibitor, right? How can you target an important gene to cure your MPN, right? Although JAK2 gave a lot of information to how disease is happening, but you also get side effects, right? If your drug is not specific to your mutant JAK2, right? And then it also hurts your healthy wild-type JAK2 that they coexist in the same patient, right? So that is one other problem. And the other problem, genotype, phenotype, I call it so you have additional mutations, okay? You target JAK2, but some other mutation also, you have it, that also contribute to not to be effective, okay? And then as we discussed, also politics, right? But that is a different topic. Now, so in other words, you really need to understand what is similar, what is different between your healthy stem cell versus your cancer stem cell. If you really tease it out, I think you will really find a good targets to target your MPN to achieve molecular remission. And then one thing I wanted to highlight actually, in this context, we are also working on now, we have a signature, molecular signature that we identified is at the stem cell level when they have mutation 1, mutation 2, or combinations compared to healthy, and then you come and attack those molecular signature which are presented only on mutant cells but not in healthy cells. Like this concept, many people working, but now we have our own approach, we have our target, and hopefully I can show you in the next meeting the outcome of this study. And with that, I would like to say one final thing. This is one of my famous cartoon again. So we discussed yesterday, and Professor Ruben Mesa nicely alluded that these patients, in addition to show classical MPN symptoms like increased blood count, splenomegaly, they unfortunately suffer from several oppressive symptoms, which including inflammation, fatigue, depression, night sweats, all cumulatively cause patient life lead to the poor quality of life. So one question we are addressing actually, how these mutations in blood stem cell cause poor quality of life. Like we studied fatigue, and why they get bad mood, why they are nervous, why they are anxiety, why they have anxiety, why they are depressive. So we are also working in that context, how this inflammation, how disease actually over time cause these undesired poor quality life. So that's what we are working now, and with that, I don't want to bother you, but I would like to end my talk by saying that we need more research, and MPN to me is like a hidden pandemic, right? It's like a COVID. When we have COVID, we all came together, we all left right, and then suddenly vaccine came. I think that much rapid and involvement we need from research side, clinician and patients, and I think we need to really. And I would like to invite you for a 30-day sugar-free challenge. It's not a ice bucket challenge. We should really test this in MPN along with this. And I would like to end my talk by saying, acknowledging my team. I have a small team. I don't have a very big team, but I have wonderful collaborators who provided me all clinical samples at our center and all over the world, and my funding support. With that. I would like to also take this opportunity to invite you all, save your date, and I'm organizing a MPN meeting in Switzerland next year, May 14 till 16. It's a beautiful place in Galen. Professor Ruben Mesa is one of the key speakers there, and Peter also coming, and we included a patient voice lectures like we are doing, and almost 40 speakers, 3-day meeting, heavily focusing on MPN. I would like to invite you all, please join and save your date, and thank you for listening.